name is Wanda Soto, and I am part of Group 4, and I will be doing restriction enzymes. Enzymes cut DNA at a specific sequence. They serve the purpose of defense against invading viruses. They are most commonly found in the bacterial cell. So when a virus DNA is present, these enzymes help cut the DNA segment, so the virus can no longer reproduce. There are 3 types of restriction enzymes, type 1, type 2, and type 3, but we'll be focusing more on type 2, as they are most common. Restriction enzymes always cleave at their near recognition sites. They produce small, well-defined fragments of DNA that help characterize genes and genomes. Enzymes are produced when a virus attacks a bacteria cell. To protect itself from the virus. The bacteria methylates its DNA, which basically means they add a methyl group to their DNA in order to protect itself. The bacteria then sends out restriction enzymes to cut out the virus DNA, which was left unmethylated. DNA being cut means that it can no longer reproduce and create new viruses. Using ACR1 as an example, since it is a common type 2 restriction enzyme, and it gets its name from the bacteria E. coli. The enzyme scans the DNA to find a specific sequence. Sequences are usually palindromic, which means that they are able to read the same forward and backward. A sticky end is created where the 4 bases are left without a complementary partner. These sticky ends make it easier for it to be attached to another DNA molecule that has been cut by the same restriction enzyme. In 1970, Hamilton Smith described an enzyme known as endonuclease R was able to cut bacteriophage, as shown in my previous slides, into specific cleavage fragments. So he was basically able to describe the process that I had gone in the previous slides. His major application to use it as a tool was to cut DNA fragments to make it easier to study characterized genes. A study performed by Daniel Nathans and Kathleen Dana at Johns Hopkins explained how the restriction enzyme can be used to make specific fragments of semen virus 40 DNA as SV40. Others showed that these fragments could be separated from one another by electrophoresis on a paloacrylamide gel. Down below, SV40 DNA is cleaved within nucleus, showing 11 fragments. He took concepts from Smith and was able to use the gel to be able to find specific RNA species of the SV40 fragments. The major use was as a device for recombining or joining DNA molecules from different genomes. In 1972, Janet Merz and Ronald Davis discovered that the ACR1 made cuts in a sequence that formed sticky or cohesive ends in a DNA fragment. It declared all R1 endonuclease ends are identical and complementary. Any 2 DNA molecules with R1 site can be recombined at their restriction sites. This opened a new path of being able to map a genetic and physical feature of a chromosome, since type 2 enzymes tend to be more specific in their cleavage sites.